The Krebs cycle produces reduced coenzyme and ATP. So the last time we went through the link reaction and glycolysis. So the link reaction produces acetyl-CoA, which is the substrate used in the Krebs cycle. So acetyl-CoA, which is a 2-carbon compound, binds with 4-carbon molecule oxaloacetate using the enzyme citrate synthase to form a 6-carbon molecule citrate. And then the coenzyme A is reused in the link reaction. And then to get from the 6-carbon molecule citrate to a 5-carbon molecule, decarboxylation happens, and also dehydrogenation. So hydrogen is removed and carbon is removed. So carbon dioxide is produced and reduced NAD is produced. Then to get from the 5-carbon molecule back to the 4-carbon oxaloacetate, carbon dioxide is produced, so decarboxylation, and dehydrogenation happens. So you've got NAD turns to reduced NAD, and FAD is turned into reduced FAD, NAD is turned into reduced NAD, and ATP is produced. So the phosphate comes from another molecule, so it's substrate level phosphorylation. The phosphate is added to the ADP to produce ATP. So per cycle, one cycle of the Krebs cycle, you get 3 reduced NAD, 1 reduced FAD, 2 carbon dioxides and an ATP. But it's important to remember that per glucose molecule, the Krebs cycle happens twice. So this is what happens per Krebs cycle. So you get 1 coenzyme that's produced, but that is reused in the link reaction. You get an oxaloacetate, which is reformed, and that can just be reused in the next Krebs cycle. You get 2 carbon dioxides, which is released as waste, an ATP, which is used for energy, 3 reduced NAD, and 1 reduced FAD, which is used in oxidative phosphorylation. So per glucose molecule, you'd get 4 carbon dioxides, 2 ATP, 6 reduced NAD, and 1 reduced FAD. So oxidative phosphorylation is the last stage of respiration, and it produces lots of ATP. So oxidative phosphorylation is a process where the energy carried by electrons from reduced coenzymes, like reduced NAD and reduced FAD, are used to make ATP. So the whole point of the Krebs cycle is basically to produce reduced NAD and reduced FAD for this final stage. And oxidative phosphorylation takes place in the inner mitochondrial membrane. So here is the whole process of oxidative phosphorylation. So hydrogen atoms are released from reduced NAD and reduced FAD as they're oxidized to form NAD and FAD again, which can then be reused in the Krebs cycle. And the hydrogen atom splits into protons or H+ ions and electrons or E- minuses. So the electrons move along the electron transport chain, losing energy at each carrier. You can see here there's 3 electron carriers. And the electron transport chain is located in the inner mitochondrial membrane. And this membrane is highly folded into cristae, which increases the surface area to maximise respiration because you can hold more carriers, electron carriers, and more ATP enzymes, ATPase enzyme. So the energy that is released when the electrons move down the electron transport chain is used to pump hydrogen ions from the matrix into the intermembrane space. And therefore there's a high concentration of protons or H+ ions in the intermembrane space and a low H+ concentration in the matrix of the mitochondria. So now you've got a high concentration here. What they want to do is diffuse down their diffusion gradient, or also known as the electrochemical gradient, because there's more H+ ions in the intermembrane space and less in the matrix. They then diffuse back via the channel for the ATPase enzyme to join ADP plus phosphate to produce ATP. So that as the hydrogens move down across the ATPase channel over the ATPase enzyme, that actually drives the formation of ATP. And that is called the chemoosmotic theory. And then at the end of the electron transport chain, oxygen, which is just from the blood, is known as the final electron acceptor. So oxygen binds with hydrogen ions and electrons to form water. So the equation for that would be half O2 plus 2H plus 2E minus goes to H2O. So that is why oxygen is known as the final electron acceptor. So here is a table to show just how many ATP are formed from one glucose in respiration. So there's 32 ATP molecules. So in glycolysis you get a net of 2 ATP. You also get 2 reduced NAD produced. In the link reaction you get 2 reduced NAD. The Krebs cycle you get 2 ATP, 6 reduced NAD and 2 reduced FAD. So in total you get 32 molecules of ATP per glucose. Remember that the link reaction and the Krebs cycle happen twice per glucose molecule. So that is the end of aerobic respiration.